Hello, everyone. I'd say let's get started. Let me introduce myself. I'm Steve Masidi. I'm the director of machines for Microfluidics. I've been traveling to India for the past 5 years, delivering these types of presentations, talking about our technology and mostly about how to solve these application challenges. This year, the trip got canceled, and we have Dr. Young Soo, who is the manager of technology and applications, and he's going to be presenting a really great presentation talking about our technology and some of the latest application examples that we have. Just a couple of rules. We are recording the meeting, so please take note of that. And also, we're going to keep everybody on mute just to minimize background noise. If you have any questions, though, please don't hesitate to take yourself off mute and ask. If you want to get our attention, there's a raise hand button. And also, we will be answering questions in the chat window. So Young will be presenting, and I will have the chat window open, and myself and some other members of the Microfluidics team are here to chat as well. So please feel free to interact with us as much as possible. And we really hope to have enough time for a question and answer session at the end of the meeting as well. So you can feel free to hold your questions until then as well. All right, so without further ado, I will turn it over to Young.
Thanks, Steve. Can everyone hear me okay? Right?
Sounds good.
Great. Okay. Yeah, good evening, everyone. It's unfortunately we can't really travel to meet you in person, so it's really nice to meet all of you virtually. I'm Young Soo from Microfluidics, and I want to thank you again for attending today's lab webinar. Here's the agenda for today's presentation. I'd like to start with a quick introduction to tell you a little bit more about Microfluidics. Although as you will see in a couple of slides that we do serve a variety of different industry, my focus today will be on the applications for biopharma industry. I'll talk about some common challenges or requirements during the development and production of biopharma nanotechnology applications, and then talk about why our microfluidizer technology is a unique technology and the ideal solution to address those challenges and requirements with a case study from these 4 applications: biopharma nanoemulsions, liposomes, polysaccharides, and cell disruptions. I will end my presentation by showing you our diverse product portfolio, and then be happy to answer any questions that you may have. Microfluidics is under our parent company, IDEX. We are part of the Material Processing Technologies, or MPT platform, under IDEX's Health and Science Technologies segment. Some of you may have already know or familiar with our other brands under MPT, but recently we just welcomed our 5th brand, Ceradose, to join our platform. Microfluidics itself was founded about 40 years ago, and we specialize in design and producing high shear fluid processors using our unique interaction chamber technology. We're located in Massachusetts, just outside of Boston, but we do have a global presence with thousands of processors sold to customers all around the world. As I mentioned already, we're part of the IDEX MPT platform. Our technology is used to help many industries, including the pharmaceutical and biotech industry, chemical industry, food and nutraceutical industry, as well as cosmetic industry, in both development stages and product manufacturing. We believe our customer success is our success, and we do our best to serve our customers with our extensive experience in applications, as well as machine designs, so we can provide tailored solution to our customers and enable them to achieve each of their specific processing goals. Now, nanoparticles and nanotechnologies are widely used in the biopharma industry. When talking about developing biopharma nanotechnology applications, especially drug products, there are many factors needs to be considered. A few common challenges and requirements related to process development and manufacturing are given here. First and foremost are the physical chemical properties. Among so many important properties, particle size characterization is one of them, and it is arguably the most important one. To be able to control the particle size and distribution of nanoparticles is critical since they control and affect other correlated properties such as solubility, stability, product performance such as ability to reach target sites, improve viability or efficacy, and etc. Particle size and distribution are also very important for generic drug development. Many of you may have heard and or are familiar with the Q123 approaches to demonstrate the equivalency of the test product to the reference listed drug, or RLD. So here, let me just briefly explain that. Q1 refers to qualitative sameness, which requires the same active and inactive ingredients as the RLD. And Q2 refers to quantitative sameness, which requires the same amount or concentrations as the RLD. For some complex generic products, a third requirement, Q3, which means physical-chemical similarity, sometimes may also be necessary. So according to FDA's product-specific guidance for generic drug development, many drug products, including some ophthalmic drugs, can use the much easier in vitro study option instead of going the full in vivo way to demonstrate the bioequivalence. So as part of the Q3 in vivo physical-chemical test, population bioequivalence, or PBE, based on the particle size and distribution within 13 confidence interval is a key criterion needs to be met. So for example, if you want to achieve matching PBE, you can either match the D50 particle size and span, or the Z average particle size and polydispersity index, or PDI. Depends on which particle size analyzing techniques you used. So even though for some generic development, even though in vivo test is still required, it is likely that you still need to do the in vitro study, which means you still need to match the particle size and distribution. So you can see here that matching particle size and distribution is very important for certain generic drug formulations. This clearly explains why the ability to choose to achieve a desired particle size and distribution is crucial when considering the right manufacturing technology. So the next requirement is that the selected technology should be robust enough and be able to produce repeatable results to ensure batch-to-batch consistency. The choice should also be future-proof in terms of scalability, which means can be easily scaled up for manufacturing with guaranteed scale-up results. Another requirement, or rather benefit, is the process should also be able to improve or lead to either downstream or adjacent processes. For example, injectables and ophthalmic drugs require sterilization to remove bacteria. Usually, autoclave is a commonly used sterilization process. However, many active ingredients are heat labile, which means you can't really use high temperature to do the sterilization, so which means autoclave is not feasible in that case. So a simple and economic alternative way to do this is by passing the product through sterile filters. Those filters are made of membranes, have rated pore size of about 0.22 microns or 220 nanometers, which means the majority of the particles in the product should be smaller than that size to ensure an efficient filtration process. So this again tied back to the requirement that you need to achieve a good particle size and size distribution, which means generating size controllable nanoparticles is very important. And last but not least, the manufacturing equipment needs to allow the entire manufacturing process to comply with the regulatory requirements such as the cGMP requirements. Many processes, especially pharmaceutical manufacturing processes, also require that the equipment is capable of running CIP, which is clean in place, and SIP, which is sterilization in place processes. So microfluidics technology can actually address all these challenges and requirements from development to production, with its unique advantages in achieving target processing results and guaranteed scalability. The uniqueness of the microfluidizer technology is that the microfluidizer processor ensures that every milliliter of material gets the same high shear treatment, regardless if you are processing a batch of a few milliliters or at thousands of liters per hour. The principle of the microfluidizer technology and how the system works explained in this diagram here. Product enters the system via the inlet reservoir and is pulled into a constant pressure pumping system, which pushes the material through a fixed geometry interaction chamber at pressures up to 30,000 psi, where the material experiences constant high shear rate and impact forces. So all materials inside the chamber receive the same treatment to achieve consistent and reliable results. After passing through the interaction chamber, the product is effectively temperature controlled and collected. We offer two different interaction chamber designs. As you can see here, showing in the top graph, this is the Y-type chamber. The Y chamber has two microchannels, and the sample impinges on each other after passing through the microchannels. The impingement lead to intensive micromixing, and so are ideal for liquid-liquid dispersing. Therefore, the Y-type chamber is ideal for preparing nanomulsions, liposomes, polymer nanoparticles, and etc. The interaction chamber showed in the bottom graph here is the Z-type chamber design. The Z chamber only has one microchannel, and the material impacts with a solid wall after exiting the channel. The Z chamber is ideal for processing samples contain solids in the case of various solid suspensions and dispersions, also include cell disruptions. As I mentioned already, that a unique advantage of the microfluidizer technology is its efficiency and uniform processing. This is the result of constant processing pressure achieved by the combination of intensifier pump and fixed geometry interaction chamber. As shown here in this graph, you can see the actual pressure profile measured for the microfluidizer processor, as shown as the green curve. So you can see as the plunger inside the intensifier pumps moves forward to pressurize the sample, the process pressure quickly reaches to the target pressure, or the set pressure, which in this case is 30,000 psi, and stays there for the majority of the cycle. The zero pressure period occurs when the plunger retracts and no sample is moved and processed during this time. So this period won't affect or lead to any interference with the processed material or results. Whereas in the case of conventional high pressure homogenizer with valves, the gap between the valve and the valve seat vibrates when the piston is constantly pumping the materials through. Although the tiny vibration is exaggerated in this animation here, it can actually cause great variations in process pressure, as also showing this graph here, as represented by the red curve. So you can see here, in this particular case, the process pressure can drop as much as 50% and only stays at the target pressure for a fraction of the entire process cycle. That means the sample was processed under many different pressures or shear rates, and you can tell the process is nowhere near uniform processing. The resulting particles usually show slower size reduction speed and with very broad distributions as compared to the much faster size reduction and tighter distribution achieved by the microfluidizer processor. As you can see from the graph shown here. So the sample processed through microfluidizer processor, which are the three green curves here, all showed small particle size with very good distribution, even after just one path. whereas the sample processed through high pressure homogenizer under identical condition, which means in this case the exact same process pressure, temperature, and for the same number of passes, showed as all these red curves here, all have large particle size and wide distribution, as indicated by either the bimodal or multimodal distributions. So even after 5 passes, the distribution was still bimodal, and the particle size was much worse than just 1 pass after the microfluidizer processor. This leads to my first application, pharmaceutical nanoemulsions, and I will use a few examples to demonstrate the use of our technology in this application. My first example is producing ophthalmic emulsion, and specifically in this case with difluprednate as the active ingredient. Difluprednate is a steroid, and it's used to treat eye swelling and pain after eye surgery. The emulsion is actually formulated with castor oil as the carrier oil. It is currently available on the market as Durezol as a brand name and is marketed by Alcon. So the goal in this study is to develop a process to replicate particle size and distribution of the RLD, as shown in both this graph and the table here. I want to point out, so you can see from the table, this particular emulsion does not only have small particle size, but also have very tight distributions, as indicated by the PDI, or generally below 0.1, which obviously would require a very robust method for processing this emulsion. To optimize the process, we tested several different process parameters as well as concentrations within the formulation. So I will walk you through the effects of changing individual parameters quickly, just to give you an idea on how to use those results during your process development and to help you reach the target particle size and distribution. I want to point out, since the sample processed through the microfluidizer processor for multiple passes is very easy and was essentially done for all tests, so therefore the effective number of passes were included when evaluating all other parameters. So the first factor we'll look at is the selection of induction chamber. Since we do have multiple options for both chamber types and size of the microchannels, as I mentioned before already, the Y-type chamber is ideal for emulsions. So we'll look at two effects from two different Y-type chambers. The difference between the two chambers that they have different size of the microchannels, which means they generate different shear rates at different pressures. So you can see from the processing results shown in this graph here. Let me just explain the graph real quick here. So the particle size is plotted against the left y-axis here, represented by the blue color. The distribution, or the PDI, is plotted against the secondary y-axis on the right-hand side here, and are represented by the green color. The two bars here represents the target particle size and the target PDI ranges. So from the results, you can see that the two Y-type chamber tested yielded similar particle size, but Y chamber 2, represented by this dashed line here, was able to achieve smaller overall PDI, which, as I mentioned before, that this may be a very important factor to consider, because in this case, the emulsion does require a very tight distribution. So we selected Y chamber 2 for further testing. So the effective number of passes was as expected. As you can see that in general, both particle size and distribution decreases with increasing the number of passes. We then look at the effect of varying process pressure with other parameters fixed. The effects are plotted here again in this graph. You can see that the pressure affected the particle size more than distribution, as the PDIs were all similar, but particle size were more separated from each other. So what's interesting in this case, as you can tell from the graph, is this particular formulation did not respond very well to high pressure, and And actually, the particle size was inverse proportional to pressure. So higher pressure resulted larger particle size. So this may also because that the emulsion was overprocessed at higher pressure. Overprocessing sometimes can happen if you put too much energy into the system during the process, which can lead to re-agglomeration or recoalescence of the droplets. So therefore, the results here suggested that this emulsion may need to be processed at medium or lower pressure. Temperature was the next parameter studied. This graph shows the effect of temperature, and you can see another interesting finding from the results shown here. So the formulation is indeed very sensitive to temperature. So however, temperature seems to have nonlinear effects on particle size and PDI. As increasing temperature, that's represented by the three curves here, initially decreased both particle size and PDI. However, at higher temperature, the results showed opposite effect. Low PDIs within the target range was achieved, which is good in this case, but particle size went back up and was above the target range. So given that achieving smaller PDI may be more critical and difficult for this emulsion, process at higher temperature may be necessary. So finally, we look at the effect of varying concentrations without changing the compositions. So using a relative concentrated formulation is often possible, especially for emulsions, since you can always dilute the sample or the product to reach the final desired concentration afterwards. Obviously, you need to check to make sure that the particle size and distribution do not change after dilution. And in this case, we tested and confirmed that the nanoemulsion was stable enough for dilution. So again, here, concentration affected the particle size more than PDI. And the effect was more complicated also. As you can see, when the concentration passed a certain limit, both particle size and PDI will start to change in the opposite direction. So I've showed a lot of results for each individual effect. So after considering the results from all the factors, eventually we were able to come up with two different formulations and process conditions that can match the RLD particle size and distribution very well. This example definitely showed the capability of the microfluidics technology in achieving a challenging goal for a very challenging formulation. The results also tells us that there may be multiple ways to get you to the target. And this actually gives you options and flexibility to be able to select a path or a process that meets your specific process requirement and capabilities. Before I move on to my next example, I'd just like to quickly point out here that during this study, we didn't really do like a formal design of experiments. Rather just use the effects to guide us to the final process. But you can certainly do this in a more formal way by conducting a DOE and use statistical tools such as the response surface methodology to better drive your process optimization. The second,
hey, Young, real quick.
Yep.
We just got a good question in the chat window. How does initial particle size of the emulsion, so the before processing material, impact the number of passes and the processing pressure?
That's actually a good question. So in general, our system, the first requirement for initial particle size is it should be in a good range that emulsion is stable, or the pre-emulsion, the coarse emulsion is stable. We don't want to see phase separation before passing through our system. So that's the first requirement. So after that, actually our system is actually robust enough. So usually we see the system can actually accept a very good range of pre-emulsion, ranging from several microns to several tens of microns. So that's usually yielded very similar results.
Great, thank you. Mr. Vivek Viswanathan, you have your hand up still. Would you like to take yourself off mute and respond? I don't know if that answered your question or if you have a follow-up question.
Yeah, hi. That answered my question. I was just inquisitive about the range which he rightly pointed out. Thank you.
Thank you.
No problem. All right, so if there's no other questions, let me continue to my second case study. In this case is an example of anesthetic emulsion that encapsulates and delivers propofol. The propofol is used to induce and maintain sedation. The propofol demand actually has been increased recently during the COVID-19 pandemic due to increased requirements as sedative for ventilated patients. Propofol emulsion is formulated with soybean oil as the carrier oil and is currently available on the market as Diprivan, which is the name brand marketed by Fresenius Kabi. And there are also several generic versions available as well. So let me show you quickly the processing results of propofol emulsion by the microfluidics processor. You can see from the results here, both the microscope images and the table, that the microfluidics processor can actually efficiently reduce the particle size of this emulsion. You can see from the image here, after processing 3 times through the system at 20,000 psi, you can see the droplets become very small and uniform compared to the unprocessed emulsion, which actually kind of give you, tied back to the previous question, kind of give you an idea about the size at the beginning. So when you look at the particle size data here shown in this table, you can see we quickly achieved a D50 particle size around 180 nanometers. And please note, so this measurement here are based on volume, so which means in this case, the D50 particle size represent that the emulsion, 50% of the droplets in terms of volume are smaller than this size. So although in this case, propofol is actually a stable drug and it's very stable at a high temperature, which means the emulsion can actually be autoclaved for sterilization. But processing results shown here that the D90 particle size after 3 passes, again, remember this is a volume-based results, so which means 90% of the droplets are smaller than this size, which is around 240 nanometers. This size is really close to the stereo filter average pore size, 220 nanometers. So this indicate that most droplets are small, and maybe they are small enough to so the emulsion can actually be filter sterilized, which is usually easier and a more economic way to do the sterilization.
Hey, Young, we've got another question.
Okay.
This is from Anupam Chowdhury. This is the question, and I don't really understand it, but I think it might have to do with bottom up versus top down. The question is, does it help in top to bottom approach in synthesis of nanoparticles? For example, can a 500 nanometer particle be scaled down to 100 nanometer dimensions by controlling pressure and number of passes?
Yeah, I'm not sure I understand the question either, but I'll try my best to answer it. I think it depends on the material and the formulation. So I would say in general, it's very possible, especially because our system does put a lot of energy. If the particles are liquid, as I showed you at the droplets in the case of emulsions or even liposomes, this is a lot easier because the droplets are softer and you can use the shear to break them, or use the energy dissipation to help break them down. So this is basically everything I showed. Let me go back here so that you can decrease your particle size with this technology. I don't really have the example shown here, but I can just make a quick comment here. If you're talking about solids, that varies a lot depending on the type of the material and the nature of the material. Sometimes we are very efficient to reduce a lot of the different materials, especially down to like a micron range or sub-micron range. But sometimes for some challenging formulations and materials, get down to like 100, 200 nanometers, that will be difficult for top-down method. And this is true actually for I think this is true for all the top-down method, not just for us.
Great, thanks, Young. Yeah, so I hope that answers the question. Maybe if you know, maybe after the call we could get some more information about this specific application. So Anupam, we will reach out to you after the call and see if we can answer the question better for your specific application. Thank you.
Thanks.
One more question. It's though it is recommended to use a Y chamber for nanoemulsions, can we use Z chamber as well?
So I think the quick answer is yes. There's nothing against you to use a Z-type chamber. It's just from our experience, Y-type chamber usually performs better than the Z-type chamber for a lot of emulsion, especially the oil water, low concentration emulsions, which I mean low concentration is the oil phase usually, I don't want to put a hard number, but usually around 20-30% of your total formulation. We do have seen in several cases Z chamber may outperform the Y chamber. That's usually the other way around, the water and oil emulsion, or the very high concentration, like if you have more than 40-50% of oil. In certain cases, we have seen the Z chamber performs better, but in general, we would recommend the Y-type chamber.
Thanks, Young.
All right, let me move to my next, which is the third case study for nanoemulsions. So this is actually a scaled up example, which is the largest scale production of an adjuvant nanoemulsion. The study was done in collaboration with our longtime partner, the Infectious Disease Research Institute, or IDRI. And the data was actually just published back in July this year in a special COVID-19 issue of Pharmaceuticals. The formulation of interest here in this study is IDRI's own squalene-based nanoemulsion developed as a vaccine adjuvant. Squalene-based emulsion have been proved to be effective vaccine adjuvants and have been used in several approved vaccines, such as the MF59 developed by Novartis, or is used in Fluad, which is the seasonal flu vaccine, and GSK's AS03 adjuvant used in the pandemic flu vaccines. The AS03 vaccine adjuvant is also used in several developmental protein-based vaccines for COVID-19. I've just heard this last week that one of those candidates co-developed by GSK and Sanofi moved into human trials last week. And the two companies were launching a very large phase 1-2 clinical trial. The objective of this study is to scale up IDRI's current manufacturing process in the case that they need to respond to a pandemic situation. And the requirement is they need to be able to supply 50 million total dosage of adjuvant within the time period of 3 months. So after considering all the factors, along with making a few assumptions, IDRI has come up with an initial estimate that the production scale needs to be able to process 1 million doses daily in terms of batch size in order to meet the requirement. Obviously, the scale-up results needs to meet the criteria of matching particle size. In this case, the Z average particle size needs to be less than 100 nanometers, along with several other physical chemical properties. The last requirement is the production batch needs to be processed at IDRI's cGMP facility, which means the scale-up solution or equipment needs to comply with the cGMP requirements. So when we start to talk to IDRI about the scale-up solution, we actually found that there was still room to improve their process since it was not done at the optimum conditions. For example, the original formulation has relatively low oil concentration, which means you need to process a larger batch to be able to produce enough adjuvant emulsion. And the process would also require more than 10 passes to reach target particle size. This also translates into a much larger throughput requirement. So we help them to optimize the formulation and conditions before scaling up their process. I'm not going to talk all that details here. If you're interested, please read the published paper, or we also have an applications note just published to summarize the paper. They have more details in there about the process optimization. I'm just going to show you the optimized results here. So the optimized lab scale results was done on our small pilot machine, the M110EH microfluidics processor that can process about 1/3 of a liter of sample per minute. And you can see from the results plotted here in this graph that process after the process and the formulation optimization, only either 2 or 3 passes is enough to achieve the emulsion with desired particle size and narrow distribution. And this condition was selected for the scale-up production runs. Before I show you the scale-up results, let me quickly explain how do we scale up and guarantee the results. So when we scale up, we don't do the same thing as the valve homogenizers would do. They usually use a larger valve and increase the gap to get higher throughput. So we don't increase the size of the microchannel inside the interaction chamber, because doing that basically ruins all the parameters you develop on the lab scale. Instead, we use multiple parallel aligned identical channels. So as long as you keep the process pressure the same, the product will experience identical uniform shear and impact treatments inside each channel, and that's achieve a guaranteed linear scale-up results. Now let's look at the scale-up results here. So remember, this is the results I showed you earlier from the lab scale or the pilot scale. So when the same emulsion was processed on one of our production scale machines, in this case the M7250 machine, which can achieve about 5 liters per minute as a flow rate at 30,000 psi. And you can see from the graph that the production machine achieved excellent results under the same processing condition. The distribution curves was slightly better after the first pass, if you look at the 2 blue curves. And after the second, which is represented by the red curves, and the third, which is represented by the green curves, the results were almost identical as the curves are overlapping on each other. So with this optimized formulation and process, so remember, we cut down a lot on the number of passes and also increased the oil phase concentration, plus a few other changes around the entire process identified and improved by EDRI. EDRI eventually came to this conclusion that the adjuvant production capacity can reach 5 million doses per day, which is 5 times of their original target. And what's better is that this 5 million dose production capacity can be done with just a single 200-liter batch. And the entire process, including the pre-mixing step, processing through the microfluidizer, large-scale microfluidizer processor, and post-process sterile filtration can be completed in less than 8 hours or within just a single shift. So this example concludes my case studies for nanoemulsions, and let's move on to my next application, which is liposomes, if there's no further questions around this. Right. So liposomes are spherical lipid vesicles with a bilayered membrane structure. So they have been used as both drug delivery system and vaccine adjuvant in commercialized products. We have presented data for liposome vaccine adjuvants in previous webinar, and so my examples today are for liposomes will be on the drug delivery side. So if you're interested in our other webinars, please check them out from our website, or reach out to us for more information. In general, when using microfluidizer technology to produce liposomes, it is essentially a top-down process based on the fume hydration method. So if you're not familiar, I'll just explain real quick here. So in the fume hydration process, homogeneous lipid mixture, including the lipophilic actives, is formed first and then hydrated, usually in the buffer solution. So the microfluidizer processor is then used to efficiently downsize the liposome vesicles to achieve desired size and laminarities. In the past few years, we've been collaborating with Professor Yvonne Perry and her group at the University of Strathclyde for liposome formulations and process development with the microfluidizer technology. The first liposome formulation we looked is the liposomal amphotericin B formulation. So liposomal amphotericin B is a well-known antifungal drug product. Amphotericin B is hydrophobic, so it is encapsulated inside the bilayer region. Liposomal amphotericin B is currently available on the market as Ambisome and marketed by Astellas in the US. The objective of this study is to create a generic amphotericin B loaded liposome formulation and study the effective process parameters to be able to create amphotericin B liposomes with properties similar to Ambisome. So liposomal Ambisome formulation include high transition temperature lipids, which means the preparation, including the hydration and the entire processing, should be carried out at a temperature at least a few degrees higher than the highest phase transition temperature. So this is important for processing liposomes. As I mentioned already, the process followed the traditional lipid film hydration method, as illustrated in this diagram here. As I mentioned already, the first step is create a lipid mixture with the lipophilic actives, which in this case the amphotericin B. And upon remove the solvent, you create a film and then rehydrate it in a buffer solution. And then the hydration causes the lipid to self-assemble into multilaminar vesicles, which is big and with many bilayers, so-called MLVs. And then you use the in this case, we use the M110P benchtop microfluidizer processor to reduce the vesicle size and laminarity. We've presented the results from this study at last year's CRS, which is the Controlled Release Society annual meeting. And I'm only going to show some data here. So as you can see that when processed this formulation at 30,000 psi, the particle size was reduced to slightly over just 100 nanometers after about 5 passes. Both PDIs and Z potential reached a plateau after about 3 passes. As expected, increasing temperature did lead to some improvements to the particle size and distribution. As you can see, the high temperature results are represented by the closed circle. So compared to the open circle, you can see the particle size was smaller with better distribution. So looking at the encapsulation efficiency here, good drug loading has been achieved after just about 3 passes, and the encapsulation rate quickly reached to about 100%. So this example demonstrates that the microfluidizer technology can not only maximize entrapment of lipophilic drug, but also reduce size, produce size well-controlled liposome vesicles with homogeneous distribution. Then move on to see if we can reproduce another well-known liposome formulation that it used to encapsulate hydrophilic active, which in this case is the liposomal doxorubicin formulation. Doxorubicin is an anti-cancer drug and has been approved for treating several different kinds of cancers and tumors. Liposomal doxorubicin is formulated with pegylated lipids and was the first FDA-approved nano drug delivery system based on pegylated liposome technology. So pegylation helps increase the blood circulation time of the nanoparticles inside the body and potentially achieve higher accumulation of those particles at the target site, for example, at the tumors, and that leads to a better efficacy. The brand name Doxo is currently marketed by Baxter in the US. And there are several generic versions of doxorubicin liposomes are available, both supplied from Sun Pharma and Dr. Reddy's. The objective of this study is to develop a generic version of doxorubicin liposome formulation to achieve a target size between 80 to 120 nanometers and high drug encapsulation rate, in this case, more than 90%. As I mentioned already, the doxorubicin liposome is formulated with pegylated lipid, in this case, the DSPE PEG 2000 with other lipids. Another unique feature about this formulation is that doxorubicin is actually loaded after the empty liposome vesicles are created. The loading is usually done by creating a transmembrane pH or ionic gradients. The process is called active loading or remote loading and can actually achieve a very high encapsulation rate. So looking at the results shown here, processed at different pressures. So you can see both of the actually lower pressure at 15,000 psi or at medium pressure 20,000 psi yielded good results in terms of particle size, distribution, as well as drug loading. With the results achieved at 20,000 psi actually more consistent before and after the tangential flow filtration or the TFF step. So the results after TFF are represented by the open circles here. So without are the closed circle in this case. So the TFF step is used for buffer exchange. Remember, we need to create the transmembrane gradients before drug loading. And also the TFF is used to remove the unencapsulated free drug after drug loading. So empty liposome particles were able to reduce to below 100 nanometers to around 80 to 90 nanometers, and the PDI was below 0.2. Quantification of drug loading resulted in over 90% of encapsulation efficiency in all tests. So although the data is not shown here, we have confirmed that after loading the doxorubicin into the liposomes, drug loading only slightly increased the particle size. So essentially, our goal has been successfully achieved here to achieve the target particles with good distribution with high encapsulation rate. And additionally, with the process involving the TFF step, there will be no further purification step required for the manufacturing process.
Hey, Young.
Yep.
Can we pop in with a couple questions?
Sure.
Okay, can you go to your last slide, please, real quick? This question is mostly about what's Z average and what's PDI.
Okay. All right. Sorry about that. So this is a different technique. So the Z average represents an average the particle size. PDI represents for polydispersity index, which usually indicate the distribution. So these are results from a different technique I showed you earlier, which is showing you all the, if you remember, the D10, 50, D90 particle size, which are based on volume. These results are based on intensity. So both techniques actually use a laser-based measurement. So this rely on the theory based on the dynamic light scattering, DLS. So the average size is somewhat at the mathematically averaged particle size. So you can, in general, just look at that's close to somewhat of mean size. And PDI, as I mentioned, represents the distribution. So if you have a single particle size, you will end up with a 0 PDI. So there's no variation. And if it's a true heterogeneous distribution, the PDI will be equals to 1. So PDI will be between 0 and 1. Usually, most people, especially for the pharmaceutical industry, people would consider PDI below 0.2 means homogeneous particle size distribution.
Awesome. Thanks, Young. I also see, sorry, Chris Jacquin also responded in the chat window. So there's
Thanks, Chris.
Another response there. Great. And then one more question. Can this be used for techniques other than thin film hydration? So this person indicates an example of like when salts are used. So that's just kind of the remote loading versus the active loading, maybe.
Yeah, so if that's the case, the answer will be yes. So, like I mentioned, so our system essentially performs a downside step. So it is used to reduce the liposome vesicle size. So if you can use remote loading or active loading, which means you create an empty liposome first and load afterwards, that's usually achieve higher encapsulation rate. But you can actually run the process to achieve what we call the passive loading. So which means you dissolve your actives in the aqueous phase, and as you perform the size reduction, you actually encapsulate that aqueous phase inside the liposome at the aqueous core to achieve encapsulation. However, usually this will result lower encapsulation rate. This is not because of the method of the preparation technique. If you use any other technique, you will achieve this similar results. It's purely results from the physical limitation, because as you start to reduce the liposome size, the volume encapsulated inside will also be reduced, and so that's basically depends on how much drug you can dissolve in your aqueous phase. I'm not sure if this answers the question. If not, maybe we can chat more or discuss more after the presentation.
Yeah, I think this is a great question, and I think probably a lot of people who are working with liposomes are trying to eliminate this thin film hydration process because it's not great, right?
Yeah. So in that regard, I want to add one. Just wait for my next case study. I'm going to show you a different way.
Please proceed.
Okay. Great. So yeah. So my next case study actually, I think partially answered that question. It's not making the film, or it's not using the solvent, which is an improved process, because with the success of the two liposome formulation, so we continued our research to see if we can solve a well-known and also it's a very important issue across all liposome preparation techniques, which is the use of organic solvents, because so far solvent has to be used either sooner or later in the process, regardless of which method you're using to create liposomes. For example, if you use the thin film method, we have discussed extensively. So you have to use the solvent to dissolve all the lipids to form the film. Even though the solvent will be removed before subsequent liposome preparation or downsizing, it's still used there. If you compare to other methods like the bottom-up method, such as the lab on a chip method or the solvent injection method, they actually have to require the use of a lipid solution to be mixed with the aqueous phase during the liposome formation step. So this means that the solvent actually presents in the final product and needs to be removed from the final product, which is usually a more difficult process. So that's a problem. So commonly used solvents during the preparation step include chloroform, ethanol, methanol, and diethyl ether. So regardless of the method of selection, the use of solvent is not preferred due to their potential safety and health concerns. So we started to think about and work on to see if we can develop a new process to create liposomes without using any solvent. And actually, I'm very happy to report here that we actually achieved that. We've successfully developed a new solvent-free process to produce liposomes with the team at the University of Strathclyde, and our work has actually just got published a couple months ago. We also presented the data already during this year's CRS virtual meeting. So with the new, let me jump to the new process. So with the new solvent-free process, we tested both previously presented liposome formulations again, and with this target in terms of particle size, PDI, and drug loading. So the new process is illustrated here in this diagram for making doxorubicin liposomes. As you can see here, instead of making the lipid film, we just hydrated all lipid ingredients directly in the aqueous buffer. and then use a microfluidizer processor to reduce the vesicle size. So same as before, we need to use a buffer exchange step to introduce the transmembrane gradients for drug loading and remove the free drug after loading. So finally, the liposome solution will pass through sterile filters for sterilization. So the process for lipophilic active encapsulated liposomes for using this process will be very, very similar. For example, if you would create the amphotericin B liposomes, all you need to do is just to also add the lipophilic actives in this step here, mixed with the buffer directly. And obviously, you don't need to use the buffer exchange step, but you still have to perform the purification. So the results are showing in this slide here. Apologize for a busy slide, but you can see this slide shows you several different charts lined for particle size and distribution, as well as surface charge and drug loading throughout the entire processing steps. The first step is the production, which is the microfluidizer processing step to reduce the liposome particle size, and then the purification step, which is the buffer exchange and drug loading step, and after drug loading and after sterilization step. So as you can see from the different bars and dots here, the desired particle properties in terms of the particle size or PDI, and also surface charge, which represented as the zeta potential, as well as drug loading, can be achieved either by processing at 18,000 psi or 20,000 psi for either 2 or 3 passes. With the higher pressure or more number of passes, slightly resulting smaller particle size and smaller PDIs. But in all this condition, you can achieve desired size, and also you can see the particles are very stable throughout the process. The size and distribution, even the surface charge and drug loading are all stays fairly constant. And you may also notice that the changes in the surface charge here before and after a buffer exchange and drug loading. So the reason was at this stage of the production, the formulation was still in this case in an ammonium sulfate buffer, which has a low pH. So after loading, or which is done by subjecting to buffer exchange to set up the pH gradient, the pH outside the liposomes become higher. So that changes lead to the decrease in the surface charge, but you can see the surface charge stays fairly constant afterwards. We then compared the liposome created by the solvent-free method to the conventional method, which is the fume hydration method. So note here, even though the microfluidizer processor is definitely capable of making liposomes via the traditional way, as I already showed you previously with the 2 case study. But for comparison purpose here, we use the sonication method to create a small batch by the traditional way. So shown here are the results of drug loading efficiency as well as morphology of the liposomes created by both methods. And you can see the solvent-free method not only proved its capability, but also demonstrate it can result better outcomes. As you can see, that encapsulation efficiency was higher than that achieved by the traditional method. I think part of this reason was also because the selection or choice of the preparation method. The sonication method used did not generate a good liposome particles, as you can see from this cryo-TEM image here. So that's a solvent-based and processed by sonication. You can see the particle size are not homogeneous. You can see some big particles and some small ones. Whereas if you compare to the liposome vesicles achieved by the microfluidizer processor showed in this images here. So you can see the very uniform particle size are achieved after pressing through the microfluidizer processor. And you can also see that in both images, you can also see that the classic rectangular shaped doxorubicin crystals inside the liposomes. So this is due to the recrystallization of the drug after loading. And this actually confirms that the drug was successfully loaded. So this case study confirms that the microfluidizer technology is not only capable to produce excellent liposomes, but also superior than other competitive technologies. In this case, the sonication method. So we also got good results with the amphotericin B liposomes prepared via the solvent-free method, as shown here. You can see after just one pass at 25,000 psi, the vesicles were quickly reduced in both size and distribution. After 2 or 3 passes, the liposome vesicle reached target particle size, PDI, as well as zeta potential and drug loading. In this experiment, drug loading in particular actually was not affected by the process at all. Liposomes can also be easily sterilized using the sterile filters, and you can see their characterizations shown in this table here. The particle size, PDI, surface charge, and drug loading was not affected after filtration. And also the morphology of the liposome can be seen from this cryo-TM images, and it confirms the particle size characterization. So in summary, example of this 2 classic liposomal formulation proved that the microfluidizer technology is very well capable of producing a variety of liposome formulations with well-controlled particle size as well as drug encapsulations. The feasibility of using the solvent-free liposome preparation with microfluidizer processor added another important benefit to our already efficient and scalable solution. Ready to move on to my next application.
Yeah, let's take a quick question. There are a few questions that we'll get to, but I think I'd rather save them to the end. But there's one more question from Vivek Viswanathan about liposomes. So let's try and talk about that now real quick, if that's all right.
Sure.
How for this solvent-free process, how do you perform mixing in the initial phase? How do you ensure thorough interaction of lipids with a hydrophobic and poorly wettable API?
It's a great question. You can let me go back to here. So you can actually read more details from the paper, but I think the step we took to ensure the thorough mixing of all the lipid or hydrophobic ingredients is we need to perform at a good temperature. And you can see in this case, we actually did it at 70 degrees Celsius. So that's above the phase transition temperature of the highest transition temperature of all the lipid ingredients. And we need to ensure we hydrate them long enough. We did an hour mixing here. You can probably do a little bit longer, depends on the formulation. And we use a high shear mixer also to help hydrate the lipids. And also it is discussed in the paper, and I wasn't showing the results here. There's depending on your formulation, there is a limitation on how much lipid or the lipid concentration you can put in there. We tested with different formulations, and we found there were different limits. But in general, we can go very high. Some formulation we achieved as high as 80 or maybe 100, if I remember that correctly, milligrams per mil as the lipid concentration, which is usually pretty high for the liposome formulations.
Great, thank you. All right, there's a couple more questions, but I think we have about 25 minutes left before the end of the session. So I think let's, you have one more case study, right?
I have two more actually. So
two more. Okay, let's go through those and then
go through them.
Great, thank you.
Great, thanks. Okay, my next application, I think it's an interesting application. It's kind of like a unique way to use our technology. This is processing polysaccharides to reduce their molecular weights. So polysaccharides are one of the most abundant material that you can find. And technically, starch, cellulose, and many other long-chain polymer carbohydrates all belong to polysaccharides. But I'll talk about a different type of polysaccharides here today, which is the bacterial capsular polysaccharide. A lot of pathogenic bacteria commonly have a polysaccharide layer, also called the capsule, outside the cell wall. So this sugar coat usually contains antigenic proteins that can be used to make vaccines against Rhodes bacteria. Polysaccharide vaccines are a unique type of inactivated subunit vaccine. Pure polysaccharide vaccines are available for diseases such as pneumococcal disease, meningococcal disease, and typhoid fever. However, pure polysaccharides are not highly antigenic, so they're often conjugated with other proteins to form conjugate vaccines to improve their immune response. One issue with capsular polysaccharide is that there are high and non-uniform molecular weights. This usually leads to a more viscous solution and makes the downstream filtration and isolation process more difficult. To solve this issue, molecular weight of polysaccharides needs to be reduced to below certain levels, so the polysaccharide solutions are more manageable. Although there are many molecular weight reduction methods exist, both mechanical method or the chemical method. The mechanical treatment is usually preferred when comparing to the chemical process because the mechanical process does not alter the structure of the polysaccharides, so their immune response can be maintained. Microfluidics technology actually works very well for this application, as you can see from this case study here. So the goal of this study was to reduce molecular weight of 9 different serotypes of pneumococcal capsule polysaccharides to below the molecular weight of 250 kilodaltons. Here are the results after processing those different polysaccharide samples through the microfluidics processor. You can see with this yellow line here represents the target molecular weight. And you can see that the microfluidics technology was able to successfully achieve the target molecular weight for 8 out of the 9 samples we processed. And most of the serotypes only require a few passes, for example, 5 or less, to reach the desired molecular weight. Many of them even just require 1 pass. And the molecular weight were already below the target. So this example certainly is a quick example, but it certainly demonstrates the capability of our technology in this application. So if you're working in the field of polysaccharide vaccines, the fast and uniform molecular weight reduction achieved by our technology can certainly benefit you, your research and your production. And then move on to my next and also the last application in this presentation, which is the cell disruption. Cell disruption or cell lysis is the process of breaking cell wall or membrane to release intracellular fluids in order to obtain molecules or particles of interest. It's an essential step during the manufacturing of many biological products. Microfluidics technology is a proven technology in cell disruption. We work very well with many different type of cells, ranging from mammalian cells, bacterial cells, to algae and yeast cells. There have a lot of publications, and we have many customers using our technology in this application. But I'd like to share a couple of interesting applications with you today. The first example is related to molecular diagnostics. And now you may be already thinking that what does the microfluidics processor have to do with diagnostics? So let me explain and show you how cell disruption by microfluidics technology can be used in this field. The field of molecular diagnostic is definitely expanding and evolving rapidly in response to the need of a rapid large-scale testing for COVID-19. Because traditionally, if you use the whole viral culture, it is not feasible because it usually takes days, a few days for the virus to cause obvious cytopathic changes in vitro. Plus, virus isolation requires biosafety level 3 facilities, which has very limited availability in many medical centers. So therefore, currently nucleic acid based testing is the ideal and the best solution. So to perform those tests, they both basically involve some basic steps. After obtaining samples from the patient, the genetic material, either the DNA or the RNA specific to the pathogen, needs to be extracted first. And then the extracted gene needs to be amplified, which means making millions or billions copies of that DNA or genetic material. So the quantity is high enough for subsequent detection. The most fundamental and widely used amplification technique is the polymerase chain reaction method, or the PCR method. Sometimes the reverse transcription polymerase chain reaction, RT-PCR, is also used. And actually, this is the case for the COVID virus, since the viral genome is made of RNA, not DNA. So an extra step is needed to translate RNA into more stable DNA before performing the PCR process. And the transcription is done by using an enzyme, which is the reverse transcriptase. The PCR basically uses a rapid thermal cycling combined with enzyme to mimic and accelerate the natural DNA replication process in order to be able to create many new copies of the target DNA in a short period of time. There are many suitable polymerase. One of the most widely used enzyme is this Taq polymerase. It is a thermostable enzyme, so it's suitable for the thermal cycling used in the PCR process. There's also another process that uses a different enzyme, the helicase. Helicases can unpack gene, which means you don't need to increase temperature to open up the DNA. So the combination of helicase and the polymerase allows the amplification to take place at the thermal condition. So all the enzymes I mentioned here, including the transcription enzyme, they all can be grown and harvested from the bacterial cell, such as E. coli. And as I mentioned already, the microfluidics technology is a proven method that works very well in rupturing this type of cell. To demonstrate this, I'll use this example. So even though this specific example did not produce the exact enzymes I mentioned in my previous slide, but rather this enzyme showed here named organophosphate hydrolase, this is still a relevant case because this enzyme was also expressed in E. coli, and the results can certainly give you an idea on how well our technology works with bacterial cell or E. coli cell. Because we're usually very efficient and can achieve over 90% cell rupture rate after just one pass. As you can see from the microscope images here, the cells are completely ruptured after processing. And also, if you look at the product recovery for the enzyme here, 1 or 2 passes at medium pressure, which is 20,000 psi in this case, give the highest enzyme recovery. With 2 passes, achieve the best overall results. And increasing process pressure starts to decrease the yield, which means you may start to degrade the proteins at this point. So the optimum pressure to rupture E. coli for this particular product is about 20,000 psi. I want to make a comment here that lower processing pressure sometimes also add a benefit when you scale up your process because our production machine would be able to provide higher throughput at lower pressure. Obviously, the best condition for your specific cells and products of interest may vary. You need to perform a little bit optimization test to determine your own best conditions. Now move to my final example, which is harvesting viral vectors for delivering therapeutic genes and next generation vaccines or nucleic acid-based vaccines. Since nucleic acid normally cannot withstand physiological conditions, so a delivery system or delivery vehicle is often required. Two types of delivery system are commonly used, either the use of viral vectors or the non-viral vector-based particles, which usually are lipid-based nanoparticles. The microfluidics technology can actually be used in preparing both delivery systems, but I'm going to talk about the viral vectors since this is related to cell rupture. We have other literatures and references for lipid-based nanoparticles, and we can share more with you afterwards or later on. So let's focus on the viral vectors here. So the vectors of interest here is the adeno-associated viruses, or the AAVs. They're very popular viruses and has actually been used in approved therapies. AAVs are commonly grow in either mammalian cells or insect cells. So regardless of what host cells are used, cell rupture is required to release the virus because they won't be secreted by the host cell. So the objective of this study is to compare the AAV vector recovery efficiency between two different methods. One is the traditional freeze-thaw method, and the other is the use of the microfluidics technology. The condition used in this test is the virus were expressed in mammalian cell, which is the HEC293 cells here, and suspended in a buffer before cell rupture. The freeze-thaw protocol was carried out by 5 cycles of freeze-thaw cycles involving freezing the cells in the dry ice isopropanol bed and thawing them at 37 degrees. The process condition for the microfluidics processor was 1 pass at 4,000 psi, which is not a super high pressure, through a 200 micron induction chamber. So you can see here, because the mammalian cells are easy to rupture, you don't need a super high shear rate. So we use the combination of a slightly larger microchannel and a lower pressure. But this is enough to rupture the cells, as you can see from the microscope picture here. After just the 1 pass, almost all the cells were completely ruptured, even at this lower pressure. And when you compare the viral titer results via the ddPCR analysis, as shown in this graph here, you can see amount of AAV harvest using the microfluidics technology is significantly higher. In this case, it's about 50% higher than that obtained through the traditional freeze-thaw method. Also, the freeze-thaw method is a very tedious method, and it took about 5 hours to complete the entire test with same amount of sample or cell or cells. But the microfluidics processor was done in less than 1 hour, including all the preparation and sanitizing step. So this is like 80% reduction in the processing time. And actually, with this this example conclude all my examples or case studies that I'd like to share with you today. I could go through another quick slide before we pause for questions. That sound good to you, Steve? All right, so before I conclude my presentation, I just want to show you quickly our product lines here. The top row here showed our different lab or benchtop units. We have a good selection, either with a unit can process very low volume with this LV1 system. It can process samples as low as a couple milliliters, with a few other selections that with different options that would suit your specific requirement. Down here shows our pilot and production systems. As showed you before, this is our small pilot system. This is our latest offering. It's a large pilot / production machine, which is the M815 processor, and this is our M700 production system. So all of our pilot production system, we have the our biopharma version that are capable of running cGMP processes. So these are highly customized machine, and we have many different options to provide you from a very basic pharma grade machine, all the way to a full aseptic version. We also have a CGMP version of our lab unit, which is the M110P biopharma unit. So with this unit, it's ideal for manufacturing a small batch required for the clinical trials. So finally, I hope through my today's presentation, you were able to know more about microfluidics and our technology. And with all the examples I shared with you, I hope I proved myself that our technology is the ideal solution for many pharmaceutical nanotechnology applications. And with that, I'd like to conclude my presentation. I would like to thank everyone again for staying late and staying through my long presentation. And I'll be happy to answer any further questions that you may have.
Awesome. Thank you, Young. And we do have a few more questions. Before I get to those, I just want to let everybody know that we are going to be sending out a survey. We're always trying to improve these webinars, and your feedback is very much appreciated. It will be sent within an hour. It should only take 3 minutes, but your feedback is very important to us. So thank you. So yeah, we'll stay on for about 5 more minutes. There are a couple more questions that we'll get to. If you have any more questions, please don't hesitate to either put them in the chat window, or you can always email Young or Daljit or me, and we will make sure that we get you in touch with the best person to answer these questions. We have a very, very good and important partner in India, Trident Equipment, and they are an excellent first pass for a lot of these questions. They've been our partner for a long time. So that you might be hearing from them as well. Great. So questions. There was a good question about graphene. Can this technology be used for graphene exfoliation?
Absolutely. Yeah. So we fall into this mechanical exfoliation process, and with the uniform shear forces and impact forces we generate through our system, we are very efficient in exfoliating graphite to achieve especially a few layer graphene, not too much for a single layer. I would say rather a few layers. And we have done a lot of work, and we have actually published a paper. We can send more information to you later. We also have a applications note that summarized that paper. Yeah, we can definitely help in that application.
Great. There's another question here about yeast cell lysis. How is that different than bacteria?
Great question. So yeast cells are in general tougher cells, so they usually require higher forces to break them. So usually for yeast cells, we have some results I didn't show here. Usually we require process at the highest pressure, which usually require at 30,000 psi for multiple passes. Yeah, we have some data. We have applications notes we can share with you. We have some good data. They're a little bit tougher, but we can do it.
Great, thank you. This is a question that you kind of talked about at the beginning about process parameters. So it says, apart from pressure and passes, what other process variables contribute to globule size distribution?
That's a great question. So I would say actually, I want to say it's equally important for process parameters as well as formulations. So the process parameters related to our system, pressure, selection or type of induction chamber, number of passes, temperature. That's usually the most important parameters. But also in my case study, I showed you changing the concentration can often lead to a bigger change or a big change in the process results or global size. So that's a lot many factors or parameters in there. The choice of the surfactant, if you're preparing an emulsion, and concentration, and the ratio of the oil phase to the surfactant. That all is going to affect the results. Just one more quick comment is we discussed about the pre-processing already. Although I don't think the particle size is truly critical, but a good premix is also important.
Okay, great, thank you. I have two more questions. Let's do these two real quick, and then we'll sign off. Cellulose. You want to talk about cellulose real quick? It's kind of the same answer as graphene, right?
Yes, it's similar. So it's we can do cellulose very well. That I would say cellulose, it's largely dependent on the type of the cellulose and also pre-treatment. You can read a lot of publications that pre-treating cellulose a lot of times will help with the defibrillation process. So we do that very well. We can we have some studies, we have some results that we can share with you guys if you're interested.
Yeah. Yeah, I'll leave that to Daljit. We'll take the lead on that, please and thank you. And the last question that we're gonna respond to, and then we'll wrap it up, is 40,000 psi. Do we work at that pressure, and is there any benefit to that?
That's a good question. So right now, no, our max pressure is 30,000 psi. We have done 40,000 psi before, many years ago. So what we found was it's not that beneficial in terms of achieving better results. You maybe get slightly improvement in the results, but considering all the effort you have to put into the machine, because once you're at 40,000 psi, now all your parts has to be a higher pressure rated parts. That's going to increase your cost of ownership, and that's so the return of the processing results do not match. And also, I know some competitors or competing technologies, they offer system goes up to 40,000 psi. So when we took a closer look, for example, I believe, which is the system of the constant, the sense that constant pressure system, they go up to 40,000 psi. But when we take a closer look and compare their system to our system, we found even at 40,000 psi, they were not as efficient as us, even at 30,000 or maybe lower pressure. So because of their system design, they're less efficient. That's why they need to go to a higher pressure. And remember, the pressure energy you put eventually will change to heat to the system. So the higher the pressure, the higher the temperature increase you will see in your sample, which is not ideal for many applications, especially for the biological products. So we're as, or if not better, we're as efficient as even lower pressure. So that's a win-win. You can get a higher throughput for larger scale. So no, we don't believe we need, you need, or we need to provide a system goes to that pressure.